All right, let's jump right into this explainer. Today, we're taking a fantastic documentary-style voyage to uncover the hidden, autonomous living factories that make up every single living organism. We're going to play the role of microscopic documentary directors, mapping out exactly how the fundamental building block of life operates, just like a highly coordinated manufacturing plant. So, how do we even study something we literally cannot see? Well, let's start with scale and origins. Scientists generally accept that life kicked off in water, specifically small pools with wild changing environmental conditions, kind of like extreme hot springs. In fact, if you look at places like the Puga Valley in Ladakh, the boiling hot springs there actually mimic the early Earth from about 3.5 billion years ago. Around these springs, calcium carbonate deposits rapidly formed, and researchers think these deposits might have sheltered early organic molecules. Basically, they formed the very first protective boundaries, which was the birth of the first cells. Flash forward to 1665. A scientist named Robert Hooke used a rudimentary self-designed microscope. We're talking just 200 to 300 times magnification here. He was looking at a thin slice of cork, discovered these little box-like compartments, and coined the term cells. When you see human cheek cells next to the rigid brick-like cells of an onion peel, it really drives home the point that the cell is the absolute fundamental unit of structure and function for all living things. But why did it take humanity so long to find them? Well, it comes down to a tiny number, 0.1 millimeters. That is basically the absolute limit of resolution for the human eye. If two tiny dots are closer together than 0.1 millimeters, our eyes simply blur them into a single point. The breathtaking 3D architecture of the organelles we're about to explore exists in a space far, far smaller than that, usually measured in micrometers and nanometers. That's exactly why we need high-powered electron microscopes to see the fine details of our cellular factory. Section 1. The factory walls. Let's breach the perimeter and check out the physical boundaries that define a cell's individuality and protect its internal operations. Zooming in, the cell membrane is impossibly thin. We're talking just 7 to 10 nanometers thick. It operates based on what's called a fluid mosaic model. Imagine a lipid bilayer. That's 2 layers of special fat molecules with proteins embedded right into them. And because these molecules can move sideways and rotate, the wall is actually fluid. It's constantly shifting. Those embedded proteins, they act as the factory's security gates. They make the membrane selectively permeable, strictly controlling what enters and exits, using processes like osmosis to move water from high to low concentrations. Now, animal cells rely entirely on this flexible, fluid cell membrane to move around. But plants, fungi, and bacteria require a totally different architecture. They possess an additional, rigid outer layer called the cell wall. Think about it. Since a plant is anchored in one place and has to withstand environmental extremes without moving, it absolutely needs this rigid wall for structural support. A flexible membrane alone just couldn't handle it. Section 2, the production floor. Moving past those security gates, we step right onto the bustling factory floor, which is filled with a semi-fluid, jelly-like substance called cytoplasm. Check out this intricate network of fine, interwoven strings. This is the cytoskeleton. In our factory, these act just like steel support beams. Actually, scratch that. They're more like a combination of support beams and internal conveyor belts. Visible only under an electron microscope, the cytoskeleton provides structural support, maintains the cell's 3D shape, and creates these dynamic pathways for materials to be transported seamlessly all across the production floor. And this network sets the stage for the actual production line, operating in a beautifully coordinated 3-step process. First, proteins are assembled by tiny workers called ribosomes. Second, they move into the endoplasmic reticulum, or ER, which spreads out like a massive network of folded membranes. The rough ER, which is studded with ribosomes, synthesizes proteins, while the smooth ER handles fats and hormones. Finally, these products are sent over to the Golgi apparatus. Picture stacks of flattened sacks that act exactly like a cellular post office, modifying, sorting, and packaging the materials into little vessels for shipping. Section 3, Energy and Cleanup. Look, you can't run a massive manufacturing plant of this complexity without a dedicated powerhouse, and you definitely need a robust sanitation crew to handle the waste. Enter the mitochondria, the powerhouse. Notice the smooth outer membrane, but see how the inner membrane is folded into these complex finger-like projections called cristae? Those folds massively increase the surface area for chemical reactions. This is where glucose and other molecules are broken down during cellular respiration to produce ATP, or adenosine triphosphate. ATP is the literal raw currency that powers almost every single activity inside our microscopic factory.
Meanwhile, waste management is handled by the lysosomes. These single membrane-bound sacs act as the cell's dedicated sanitation department. They are packed with incredibly powerful enzymes that can break down unwanted proteins, carbs, fats. They can even completely destroy damaged or worn-out organelles. They basically digest these broken components and recycle the raw materials right back into the cytoplasm. It's the ultimate green recycling program.
Storage logistics also differ pretty drastically depending on the type of factory. Notice how mature plant cells feature one massive central vacuole surrounded by a single membrane. It stores water and cell sap, maintaining this heavy outward pressure that keeps the plant cell incredibly firm. If a plant doesn't get enough water, this vacuole shrinks and the whole plant visibly wilts. Animal cells, on the other hand, just have much smaller vacuoles used for temporary short-term storage. Section 4, the master blueprint. We are now entering the most heavily guarded area of the cell, the CEO's office, the prominent double membrane-bound nucleus. The nuclear membrane has these highly specialized pores allowing for tightly controlled material transfer. Inside, we find chromatin. During normal operations, it visually resembles an entangled mantle of thread, kind of like intricate embroidery yarn. This chromatin is made of specific proteins and, of course, DNA. The DNA holds the exact genetic inheritance codes and blueprints that dictate every single structure, product, and function this factory will ever produce. So just how absolute is DNA's control over the factory? Well, a completely mind-blowing 2010 synthetic biology experiment by scientist J. Craig Venter proved this perfectly. His team chemically synthesized an exact computer-programmed copy of a simple bacterium's DNA. They removed the original DNA from a host cell and swapped in the synthetic blueprint. Instantly, the host cell began operating, growing, and dividing completely under the instructions of the new synthetic DNA. I mean, the blueprint truly is the ultimate master controller. Section 5. Expanding the factory. Knowing how flawlessly engineered this system is, how does this microscopic facility ensure its long-term legacy and repair damage? It does this through a highly controlled, orderly cell cycle called mitosis. When the cell is ready to divide, that tangled chromatin we talked about organizes into distinct rod-shaped chromosomes. Mitosis then perfectly duplicates this DNA and splits the parent cell into two genetically identical daughter cells. Each new cell gets the exact same genetic info, the exact same number of chromosomes, ensuring the organism's instructions are perfectly maintained as it grows. Take a second to let this staggering number sink in. Hundreds of billions. That is the number of cells replaced in your body every single day through division. That's almost 1% of your total cellular makeup. You are quite literally regenerating a fraction of your biological factories every 24 hours. Constantly growing, repairing tissues, and replacing dead cells that have reached the end of their naturally programmed lifespan. You are the product of trillions of these hardworking, highly ordered factories. Normally, when animal cells bump into neighboring cells, they naturally stop dividing. It's a brilliantly designed safety mechanism called contact inhibition. But when cells lose this control, they divide uncontrollably, leading to the formation of tumors, which we know as cancer. Understanding this beautiful, complex, microscopic factory isn't just about learning biology. It's about unlocking the very secrets woven into the fabric of life, waiting for us to cure them. So what other mysteries are hidden in that 0.1 millimeter space? I'll leave you to ponder that until our next explainer.